Hi, I am Alejandro de Santiago, and I am a PhD candidate in the Institute of Bioinformatics at the University of Georgia, where I study the microbiome of microbial invertebrates that live in marine sediments. Marine ecosystems harbor diverse animal life and complex communities of microbial organisms. The bacteria colonize both ocean sediments and living organisms. The community of microbes that colonize living organisms, such as coral or fish, are typically referred to as a microbiome. The animal's microbiome can impact their ability to survive toxic habitats, such as hydrothermal vents and methane seeps. In deep seas, hydrothermal vents and methane seeps can produce a lethal amount of hydrogen sulfide and heavy metals. However, some bacteria are able to protect their hosts from such toxicity and provide them with necessary nutrients. For example, large tube worms that live in hydrothermal vents lack a typical digestive system. Instead, they have an organ called the trophosome that harbor a bacterial community that is able to break down the toxic hydrogen sulfide into non-toxic sulfur through a process known as sulfur oxidation. Even microscopic roundworms, known as nematodes, harbor bacteria that live in the gut and on their exoskeleton. Nematodes are a highly diverse group. They are present in almost every habitat and play an important role in maintaining healthy ecosystems, ranging from agricultural systems to marine sediments. In marine sediments, free-living nematodes and their microbiome play a direct role in regulating biogeochemical processes and decomposition. Despite their importance, they are extremely understudied. There is some evidence that nematode-associated bacteria might be common. Mouthless nematodes, known as nematids, thrive in sulfur-rich environments. These worms are completely covered in a single species of sulfur-oxidizing bacteria. Remarkably, nematids that have their symbionts removed are no longer able to survive in these habitats. This suggests that these bacteria play an important role in detoxifying the worm's immediate environment. Also, recent microscopic images have revealed that bacteria live in the gut, oral cavity, and on the exoskeleton of various nematode species called oncolamids. However, there are no genomic studies that have tried to identify the functional potential of the oncolamid-associated microbes. Therefore, I am sequencing the genomes of the worm's microbial community to better understand the interactions between the nematode and their microbiome. As part of my research, I am developing a computational workflow that can be used to rapidly study the microbiome of other microbial invertebrates, such as copepods and tardigrades, otherwise known as sea bears. Understanding the interactions between animals and bacteria is necessary in order to characterize the complex interactions that occur in ocean sediments.